cytosine and guanine. Pyrimidine you have cytosine and thymine. Cytosine and uracil in the term of in RNA. Okay. So you compare first to base. Why do we call this a base? First we must understand why we call this a base. எங்க இருக்கு இங்க அதுவே பாஸ்போரிக் இது மூணு இல்லையா base deoxyribose and phosphoric. So why is this called a base? So what is our definition of a base? Very good. It accepts electrons, right? Does it accept electrons or does it give electrons? Base gives electrons, right? So for example, why is ammonia called a base? Ammonia there is no hydroxyl ion, right? But we still call it a base. Why do we call it a base? Because when you put it in water, right, there are lone pairs of electrons and that takes the H plus from water and releases a OH minus. So you will see that anything that has lone pair of electrons that can accept, that can give electrons, we will call it a base. So otherwise then we can call this a base because there are lone electrons on this nitrogen and this nitrogen, and therefore we call it a base. Now sugars பத்தி உங்களுக்கு தெரியும் இல்லையா? So என்ன sugars எல்லாம் do you know about what kind of sugars do you know? Glucose and fructose, right? So sucrose is made up of one molecule of glucose and one molecule of fructose, correct? So the sugar would so இப்போ நீங்க பாத்தீங்கன்னா fructose is a 5-membered sugar. Glucose is a 6-membered sugar. So what shall we call them? For example, a 5-membered ring, what will we call it? Penta. So other pair we can call it pentose. So so fructose is a pentose. Glucose is a hexose, right? So in the body, depending upon the arrangement of the OH groups, there can be different sugars. So one of the sugars that resembles fructose is called ribose, right? So this is a ribose. So ribose is a pentose. So the numbering pattern, you will see it as 1, 2, 3, 4 and 5, right? So you can see that 1, 2, 3, 4 and 5, correct? So you will see that ribose is a sugar, right, with a pentose. And if you would have heard of certain other names like dehydrate. What is dehydrate? Emulsion of water. So deoxyna, removal of oxygen. So for ribose, ribose will have a OH here, right? So if I remove that oxygen, what should I call it? Deoxyribose. That's why it is called deoxyribose. That's where I remove that oxygen from the second position. So I will call it 2-deoxyribose. Correct. So now this OH and this hydrogen can react. There can be a dehydration reaction, and therefore you can form a bond. So this is the. So the first slide you told me about esterification, right? What is the esterification reaction? Alcohol and carboxylic acid. So that is basically an organic ester, correct? So ademari you can also see that you can form inorganic esters. So inorganic esters enna pannalam? We can take an alcohol and react it with mineral acids, right? So that's exactly what nature does. So this is a alcohol, yeah, CH2 reacts with phosphoric acid, which is a mineral acid. Takes off water. Remember, number esterification reaction also we take off water, right? Ethanol plus, what is the other reactant? Ethanol plus acetic acid gives you ethyl acetate plus water. So adhe you can now take this deoxyribose CH2OH reacted with phosphoric acid and then remove the water and then you get a phosphate, right? So this is an inorganic phosphate. For example, it is known, actually if you go, you can chemistry wise you can actually get triethyl phosphate. Okay, you can buy it from. So what is triethyl phosphate? All this H, all these three H's are replaced by ethyl groups, right? So OCH2, CH3, OCH2, CH3, OCH2, CH. So we can call it triethyl phosphate. So this is, so now this is a, now you can see what is the name of this compound. So this is called a nucleotide. So what is the name of this compound? How shall we name it? Now we have to name it to take into account all the three parts of the molecule. So we will first take deoxy adenine, right? So we will say this sugar is the same everywhere, right? So this is going to be deoxy. This is going to be different. How many types of bases do we have? Four, right? Right? Adenine, thymine, guanine, and cytosine. So we will see that this is deoxyribose, adenine, right? So adenine, deoxyribose. Because of the P is phosphate, M is monophosphate. So this is how we will name it, right? We will call it deoxyribose adenine monophosphate, right? So this is a nucleotide. So we will go to the next thing. So each of our DNA, right, contains this. This is our basic material, right? Next is we have to, our DNA is a huge molecule, right? So how is this chemistry? In the chemistry and in the groups how can you make a huge DNA molecule, right? In the molecule we cannot have all the information for such a complex being. So so we can do another chemistry. What can we do this another chemistry? Chemistry from a small molecule, if I want to make a big molecule, polymerize. Very good. So you make use of polymerization. What is the essential condition for polymerization? Okay, we have to make chains. That is, there should be at both the ends of the molecule. At both the ends of the molecule, you should have a double bond. Very good. So you should have a reactive group. Are you able to follow? Both the ends of the molecule, you should have a reactive group. As he very rightly said, a double bond will make that. If example of a polymer with double bonds, polyethylene. The soles of the shoes and chappals enough use for that. No, you can't use polyethylene, right? It will rub off very fast. Or do you use very hard material? What is it called? Velcro is there. In the soles, polystyrene. Have you heard of that? Polystyrene. Okay. Don't worry too much about it. Polystyrene the CH2 polyethylene CH2 double bond CH2. So one of the hydrogens might become phenyl. Benzene ring, that is polystyrene. One of the hydrogens you take, so phenyl or benzene, CH, double bond, CH. This is polystyrene. So the essential part for any polymerization reaction is that it should have two double bonds, two reactive groups. Double bonds support on. Okay, it has to that two in the same molecule, in the same atom which is bonded, right? There should be two reactive groups. So other number in the compound in this compound. Don't think about do we have in any part of this molecule two reactive groups? No one. எல்லாருக்கும் புரியுது அந்த क्वेश्चन எல்லாருக்கும் புரியுது அந்த क्वेश्चन இந்த மாலிக்யூல்ல வந்துட்டு ஏதாவது இந்த இதுல two reactive groups இருக்கா ஒரே மாலிக்யூல் very good in the oxygen O minus can react with another OH of the other sugar, correct? Very good, excellent. So now you got it. So this is exactly what it does. So in the so you have the CH2OH here. This reacts with this oxygen. There is another OH here that reacts with this. So this goes on building. So we can easily make that is in the CH2OH. This is that CH2OH. Ingo oxygen. This oxygen can react with this OH. Correct? And then we can make this CO. So we can make a polymer. So polyethylene how many monomeric units are there? Two. No. Two reactive that is CH single bond CH single bond CH single bond other on the end of protocol. What is that end? How much is it for polyethylene? About 100,000. That N is equal to 100,000. So that is correct, yeah, because ethylene is a gas, but polyethylene is a solid, right? So you have to have large. So very similar to that. What is N in our DNA? Are you able to understand the question? No? Okay. So now we have said that this is the monomeric unit, right? This is the monomeric unit. This is the polymeric unit. That is goes on repeating. Only thing that will be different is the base. The bases will be different, right? All others will be everything is the same. In our DNA, the bases are different, right? So how many such polymeric monomers will for example we said CH single bond CH is N times that is 100,000. This thing goes on repeating, right? So this N is going to be our DNA. Right? That is N is going to be. So how much is N for DNA? 1 trillion is the number of cells. DNA it is actually 3.5 billion. Okay, this N. So you know that this goes on repeating, right? And this is going to be, this N is equal to 3.5 billion. And you just note it down, where we will use it later on for some calculation. So this N is equal to 3.5 billion, right? So what can we say? That our DNA is made up of 3.5 billion bases, correct? Because only it is not constant, right? Only this will be different, right? So we will say that our DNA is made up of 3.5 billion bases. This how many of you know the KVPY exam? So how many of you know about the KVPY exam? Only these people. Please go to this website. And there's one more chance. Two more chances. 11 and first year. So please do write this exam. So if you pass this exam, you will be called for an interview. And the last year, I was a coordinator, and this will be held at Anna University. The interviews will be held at Anna University. I am a coordinator. Interviews at Anna University. Please do write this exam. This is a very interesting exam. Of course, it is, I think, in one sense, biased towards CBSE, but you can't help it. But please do write this exam. You can write it at any time. If you write this exam and pass this exam, you are eligible to apply for any of these institutes. The Indian Institute of Science Education and Research at Mohali, Pune, Bhopal, Trivandrum and Calcutta. And also, if you keep up your same academic performance, you are also in the 12th, you are also eligible for this to the Inspire Scholarship. This is a 5-year, I sort of request you all to go through this websites of all these Indian Institute of Science. Because I think next year there will be another one at Tripoli. Have they put that website? I think it will take another 2 years for Tripoli to come in. So please go through this and please go through the websites also. You get it's a 5-year integrated BSMS course in physics, chemistry, maths, biology, and mathematics. Yeah. Physics, maths, biology, and chemistry. Yeah. Physical and chemistry. Only IAC has geology, earth sciences, and material science. It's a 4-year BSc course. This is a 5-year MS course. So I request all of you to go through this and go through each of the individual ones. For the first 3 years you get 5,000 rupees as your scholarship. A very, very nominal fee of 24,000 rupees a year, which includes boarding, lodging, everything, except food. And a 20,000 rupee book grant every year. And after the 4th and 5th year, you get 7,000 rupees and a 28,000 rupees book grant. Please go through this, and let me tell you that it is fantastic that by the 2nd year you can use equipment that even PhD students in normal universities can't use. I can assure you that it is all good, because I do go to ISER Mohali every 3 months. My son is a KVPY scholar. He's in the 3rd year of MS at Mohali, so I do try to go there and work. And I wish you all the best. I'm very sorry that I could not do this totally in Tamil, but if you have any questions, please do ask me, or even write to me. I will do this. We are planning to do something with our
why can't we take something with microbiology?
Oh, okay. Because microbiology, by its definition, talks about life, and viruses are still not classified as life. They become life when they infect the cells. Generally, they are not put into microorganism. Micro means micrometer. Viruses are much smaller.
Escherichia coli is known as common cells. Escherichia coli, disease is called as common cells. What are common cells? Common cells. COM.
Okay. Common salts are things that live with us. Okay. This is something that I have to sort of. There are more bacteria inside us than other cells that we have. Okay, we have more than 1 billion bacteria inside us, and all of these don't live with us, but live in our environment. So they are all called non-common, and most of them are pathogenic. Most of them. The guys who live with us are not that pathogenic, but they can become. For example, E. coli can become exceedingly pathogenic sometimes. For example, you would have heard about the dysentery or uropathogenic E. coli. Those are all confused guys.
So, some bacteria are distinguished as gram-negative and gram-positive. What are those?
See, bacteria have a cell wall, right? So the cell wall for some bacteria are made up of hydrophobic components, more hydrophobic components. See, of course, I didn't explain that when I did this. Is in that book, in that book.